Okay, let's dive into this. Welcome to The Explainer. Today, we are going to explore the absolutely fascinating and, mostly hidden world of molecular biology. We're going to essentially sketch out the inner workings of the ultimate manufacturing plant, the living cell, just to really understand the incredibly diverse and dynamic roles of RNA. If you have ever wondered how the completely static code of your DNA actually translates into a living, breathing, functioning organism, well, you are in the exact right place. Think of this as our whiteboard syllabus for today's session. We are going to quickly map out the central dogma, then look at mRNA as our messenger, tRNA and rRNA as our core builders, check out the specialized RNAs that kind of act as middle management, and finally wrap up with the ancient RNA world hypothesis. Let's move right into our first section and see how this all builds, starting right at the very foundation of all biological life, the central dogma. The central dogma is absolutely the fundamental process of gene expression. Basically, it describes how genetic information flows in one specific direction. If we stick with our cellular factory metaphor, information has to eventually become a physical product, right? You can't just have a blueprint sitting there. You need a process to turn that blueprint into the molecular machines that actually do the daily work of keeping you alive. So here is how that manufacturing plant operates in 3 sequential steps. Step 1, your DNA is the master blueprint. It's incredibly valuable, so it stays securely locked away in the executive office, the nucleus of the cell. Step 2, RNA acts as the intermediary messenger. It copies the instructions and does all the critical legwork of carrying that information out of the nucleus. And then step 3, the final output is generated. The cell produces proteins, which are literally the physical machinery making up your tissues, your enzymes, and all your metabolic pathways. Now that we have that overarching workflow mapped out, let's zoom in on the delivery system itself. Section 2, mRNA, the genetic messenger. Meet messenger RNA, or mRNA. Through a process called transcription, an enzyme called RNA polymerase reads a specific segment of the DNA blueprint and synthesizes a complementary mRNA strand. So this mRNA is our middleman. It carries the genetic code from the nucleus out into the cell's cytoplasm. But here's the cool part. It doesn't just carry a random string of letters. It organizes this information into distinct triplets of nucleotides called codons. Think of them as individual building blocks. And each codon specifies one very particular amino acid, which of course are the fundamental components of proteins. Okay, so the mRNA has successfully exported the instructions. Great. But that leaves us with a pretty massive conceptual gap. How does the cellular factory actually read this mRNA message? I mean, how do we go from just an information-carrying messenger to physically building a complex 3D protein? Well, to answer that, we have to shift our focus down to the bustling factory floor and introduce the active machinery that translates this code. Section 3, tRNA and RNA. So the crucial point here is the contrast between the 2 main workers on our assembly line, tRNA and rRNA. On one side, we have transfer RNA, or tRNA. These are short 70 to 90 nucleotide molecules folded into a really distinct 3-dimensional cloverleaf shape. You can think of them as the factory's transport trucks. One end carries a specific amino acid, and the other end features an anticodon that perfectly pairs up with the mRNA's codon. Now, on the other side, we have ribosomal RNA, or rRNA. These are the assembly line managers. rRNA combines with proteins to form ribosomes. But, and this is key, rRNA isn't just structural scaffolding. It is the actual catalytic engine that locks those amino acids together by forming peptide bonds. Now, what's incredibly fascinating about this is exactly how that peptide bond is formed. For the longest time, scientists just assumed that the proteins within the ribosome were doing the actual chemical work of building the new protein. But when we look at the high-resolution atomic structure of the ribosomal active site, specifically the peptidyltransferase center, we see something truly astonishing. There are literally no protein atoms within 18 angstroms of the reaction center. The rRNA is doing all the heavy lifting. It's not just a passive structure. rRNA is an active enzyme. It's a ribozyme, catalyzing the fundamental chemistry of life. For decades, the central dogma, DNA to mRNA to protein, aided by tRNA and rRNA, well, that was the whole story. But molecular biology has revealed a massive layer of complexity way beyond core translation. Let's look at Section 4 and meet the specialized RNAs. The genome is actually pervasively transcribed, producing an absolute plethora of non-coding RNAs that regulate the entire operation. First, we have SNN RNAs, small nuclear RNAs. These process and splice the mRNA before it even leaves the nucleus. Then we have SNO RNAs, small nucleolular RNAs, that guide the chemical modification of other RNAs. And incredibly, we have eRNAs, or enhancer RNAs. These are transcribed from enhancer regions of the genome, and they are phenomenal. They actively instruct transcriptional programs in space and time. Research shows eRNAs can modulate chromatin looping, which means they are literally bringing distant parts of your DNA physically closer together. They can trap transcription factors to boost gene expression, and they even help form these liquid-liquid phase-separated condensates. Basically, that creates temporary compartments inside the nucleus just to crowd transcription machinery together. They are the ultimate middle management of the genome. Realizing that RNA can carry code just like DNA and act as a catalytic machine, exactly like a protein, really forces us to take a step back in time. Let's move to Section 5 and map out the RNA world hypothesis. Think about it. Billions of years ago, before DNA and complex proteins even existed, how did life function? Well, the RNA world hypothesis suggests that RNA just ran the entire factory all by itself. And the evidence for this is super compelling. First, RNA can store and replicate genetic information, just like DNA. Second, as we just saw with the ribosome, RNA can catalyze critical chemical reactions, just like protein enzymes. And third, many of the core coenzymes that are totally fundamental to cellular life today, things like NADH and acetyl-CoA, are structurally strikingly similar to RNA. It strongly suggests they're ancient surviving remnants from an era when RNA was literally the only player in town. And naturally occurring ribozymes brilliantly illustrate this exact concept. We have identified chemical mechanisms of self-cleaving and self-splicing ribozymes. Basically, these are RNA molecules capable of carrying out phosphodiester cleavage and ligation reactions entirely on their own. They often use their own nucleotide bases for the catalytic chemistry. They don't even need a protein. These ribozymes give us a direct structural window into that primordial RNA world, showing us exactly how early life might have catalyzed its own replication and metabolism. So we've seen that RNA is definitely not just some passive middleman. It carries the genetic code, it structurally builds the machinery of the cell, it regulates the entire genome, and it very likely predates DNA itself. So I'm going to leave you with this to think about. If RNA is the architect, the messenger, the builder, and the manager, is RNA the true secret to the origin of life? Thank you so much for joining me on this explainer. I really hope this has completely changed the way you view the molecular factory operating inside you right now.